Thanks. Hi, everyone. So today I'm going to be presenting some work that's actually in review at the moment, and it's with my co-authors from JCU, Phil Munday and Jenny Donaldson, who you just heard from, and collaborators from King Abdullah University of Science and Technology, Professor Tim Ravasi and Dr. Heather Veilleux. So as we just heard from Jenny, coral reef species has the capacity to rapidly adjust to ocean warming if exposed early in development and across generations. But the underlying molecular mechanisms for this process are largely unknown. So my work focuses on understanding the processes behind this acclimation. Well, before Watson and Crick, before Mendel, and before Darwin's theory of evolution, there was a renowned French scientist named Jean-Baptiste Lamarck who wrote the framework for evolution, and these were concepts that were widely accepted at the time. His first law was that if you have an organ and you use it a lot, it enlarges and develops. But if you don't use that organ, it will weaken, deteriorate, and eventually disappear. And his second law was that these changes could be inherited. The most famous example of this Lamarckian evolution are the giraffes. So the more the giraffe stretches its neck to reach the leaves at the top of the tree, the more it will strengthen and lengthen, and then this long neck can then be passed on to the next generation. But by 1880, German evolutionary biologist August Weismann found out that DNA in the gonads is separate from the DNA in the rest of the body. So it doesn't matter what you do in your neck and your muscles, it can't be passed on. So Lamarck's theory of evolution quickly fell out of favor as a viable mechanism for adaptation and inheritance. Today we know that we've got the exact same DNA in every cell of our body. And encoded in the DNA are genes. And when we need to make use of the gene, it gets transcribed or copied into RNA. The gene is expressed. And then it gets translated into protein. So how do we go from a single egg, a single fertilized egg, into an embryo and then an adult with all the various cell types, tissues, and organs? How does a liver cell know it's a liver cell and not a brain cell, even though they've got the exact same DNA? Well, each cell type has its own repertoire of genes that are expressed. And this is an extra layer on top of the DNA code that's essential for cell differentiation and development. And this is called epigenetics. Now, can the environment cause this extra layer of information on top of the DNA? Genetically identical honeybees can be epigenetically different. So if given the royal jelly early in development, the queen bee will end up looking phenotypically quite different than the worker bees. Also, sea turtles, the eggs that are buried in the sand experience different temperatures depending on depth, and this epigenetically determines the gender. In a human example, during World War II, there was a village in the Netherlands that was cut off from food supply, and mothers who experienced famine during the first 10 weeks of their pregnancy ended up having babies with lower birth rate and who went on to have increased rates of diabetes, obesity, and cardiovascular disease. And this was compared to siblings who were born outside of the famine. And this shows that experience early in life can have long-term consequences. So how did this happen? Well, a study found that there were changes in methylation in genes involved in imprinting and metabolism. And what's DNA methylation? Well, it's that extra layer of information on top of the DNA. It's specifically when a methyl group gets added to the cytosine of the DNA code. And that typically causes the DNA to wind more tightly and become inaccessible, and then the gene can't be expressed. Now, can these epigenetic changes be passed on to the next generation? And this epigenetic inheritance is quite a hot topic. There have been many models and mechanisms that have been proposed. And perhaps one of the most robust examples is that of a gene that is attributed to coat color in mice. So the normal gene, the wild-type gene, the agouti, is activated during a short time during hair development and causes a little yellow fleck in an otherwise dark coat color. Now, the agouti viable yellow gene has an extra bit of DNA before the gene that can be either methylated or unmethylated. So when it's unmethylated, it's open and on, and it's represented by the white lollipops in this figure. When it's methylated, it's turned off, and there are the purple lollipops here. So when unmethylated, this IAP actually acts almost as an amplifier, causing that gene to be constantly activated, and then the coat color turns yellow. If it's methylated, turned off, it's not activated, and the agouti viable yellow gene acts more like the wild type and it causes a dark brown coat color. In some cases, you can get some cells in the mouse that are methylated or unmethylated, and this causes a mottled appearance. But can parental experience or environment actually cause changes epigenetically in their offspring? Was Lamarck not totally off the mark? Well, when mothers were exposed or given a diet of vitamins like folic acid that increase your methyl donors, it caused the mice to have that IAP to be methylated and turned off, and then that resulted in only having mice with the dark brown coat color. There were none that were yellow. So bringing this back to marine species and climate change, are there any epigenetic mechanisms specifically to allow that spiny chromis that Jenny spoke about in the last talk to transgenerationally acclimate? So we looked at 5 different treatments of that second generation spiny chromis that were exposed to increased temperatures, so 1.5 or 3 degrees above current day. And they were either controlled treatments, developmental, so they were exposed from early development for 1 generation, or transgenerational, exposed for 2 generations. And as Jenny mentioned, the transgenerational fish fully restored their aerobic performance compared to the developmental fish. So this shows that transgenerational acclimation is possible. But what goes on at the molecular level? Well, we looked at all of the genes that were expressed in these fish, and we found that there were 87 genes that were differentially regulated among these 5 treatments. And they typically had to do with metabolism, stress, and immunity, and other developmental processes. This got us wondering how. Is this epigenetic inheritance? Are there changes in methylation like what we saw in the mice? So we undertook a massive next-generation sequencing experiment where we use cutting-edge techniques, and we sequenced the entire genome, so all of the DNA in these spiny chromis. We looked at the methylome, so all of the methylation changes across the genome, and we sequenced all of the genes, the transcriptome. And we did this for 4 different second-generation spiny chromis fish. So control, developmental, and transgenerational exposed to the higher temperature, in addition to the step treatment where the parent was exposed to 1.5 degrees and then stepped up to +3 degrees in the second generation. And this treatment also showed acclimation of aerobic performance. And what we found were 2,205 differentially methylated regions that were actually associated with genes. And as you can see from the heat map, the transgenerational fish showed the most changes compared to control, and the developmental showed the fewest changes compared to control. So 51 of these genes also had differential expression, and I've put these in a table here and color-coded them based on their most likely and perhaps most basic function. So if you're only exposed for 1 generation to high temperatures, you only have 1 gene that was differentially methylated and expressed. If you're exposed more gradually across generations, we found 6 genes that were differentially regulated, and most of them had to do with metabolism. But if you're exposed for 2 generations, this caused quite a few genes to be differentially regulated, encompassing a wide range of functions. But some of these genes might actually be working in concert to affect overall changes and improvements in condition. For example, these 5 genes might serve to improve oxygen transport and circulation in the circulatory system. But which genes might play the biggest role in acclimation? So what we did is we took the individual gene expression and we correlated it to what we know is acclimating the aerobic performance. And we found that 7 genes were significantly correlated. And one of the interesting ones that I just wanted to highlight was NLRP3, which plays a critical role in inflammation. And it can be activated in response to reactive oxygen species, which we know are increased in warmer conditions. There's also apolipoprotein A4, which is involved in lipid and cholesterol metabolism. And this is actually quite frequently found in many thermal studies in fish, so it might play a very important role in responding to and adjusting to ocean warming. So just in conclusion, we've shown through this very innovative and high-throughput project that a coral reef fish can epigenetically regulate genes associated with acclimating to ocean warming. We also found that changes in methylation occurred with increasing exposure to high temperatures, and most of those differences were when the fish were exposed transgenerationally for 2 generations to the high temperatures. Finally, 51 of the differentially methylated genes were also differentially expressed, but this is only 2%. And this was quite surprising for us. We would have predicted more genes to be also differentially expressed, but this is actually being found quite common, and as the number of molecular studies are increasing like this. And it could be due to an issue with timing. So these genes that were differentially methylated may have needed to be differentially expressed early in development so that it could impart lifelong changes for the animal. And they don't need to be differentially expressed when we sampled the fish as adults. It also could be that there are other epigenetic mechanisms at play. There are a variety of things that might be going on, like RNA or transposons, and this is really opening up an avenue to pursue for really figuring out what's happening in these fish. So I've got a lot of projects that are currently on the go. So what we want to know is what's actually happening in the 3rd generation of these fish exposed to high temperatures. What's the methylation patterns? What are the gene expression? Specifically, if we put them back into controlled temperatures, does everything stay the way it was, or does it actually go back? What happens when they're exposed at different times, like Jenny was saying, for reproductive purposes? We're also wondering what happens to 2 different populations of reef fish that we know have different capacities for acclimation. Specifically, if we expose them just as adults for short-term heat exposure, like what was seen on the Great Barrier Reef last summer and this summer. And then we look at the genes that are differentially expressed in those fish. And one of the really, really exciting projects that are coming up, when we found out about the bleaching that was happening on the Great Barrier Reef, we set up a massive transcriptome analysis in which we collected 9 different species at 4 different time points before, during, and then after the bleaching last year on the reef. And we are evaluating the transcriptome. It's actually being processed right now at KAUST, and we hope to be able to eventually identify genes that might be key indicators of thermal stresses. We know some of these fish are actually more susceptible or more tolerant to warmer temperatures. And that's it. Thank you to my followers.